Director at the Center for Watershed Sciences at the University of California, Davis. Dr. Jeffrey's research program is broadly focused on applied aquatic ecology and physical processes with an emphasis on anadromous fishes across freshwater and estuarine ecosystems. His research aims to fill critical data gaps needed by fisheries and water resource managers to implement effective restoration and conservation measures for both imperiled and other native species. Dr. Jeffrey's work with a variety of partners from farmers and ranchers to agency personnel to generate novel solutions for difficult natural resource problems. Dr. Kwok, he is a senior fisheries veterinarian for CDFW, and he obtained his veterinary degree and PhD in fish virology, both from UC Davis. Dr. Kwok helps to oversee fish health throughout California. So thank you all. Let's give a virtual round of applause for our presenters. Yes, thank you. And Dr. Jeffries, whenever you're ready, please take it away.
All right. Thank you, Richard. Let me see if I can be successful in this on the first try. All right, can you all see that?
Yes.
Okay,
yes.
Great. Yeah, it's funny. Every platform has a different thing. I can't actually see any of you anymore. We will go from here, and I will go blind and hope you're still there. It's so cool to be here, and I appreciate it so much. And especially looking through the names, I recognize some of the names in the group here, both from work. There's fish people in here, there's agency people, there's teachers who I've worked with in the past, and so it's pretty awesome to be able to have this part of my life kind of intersect with other parts, and I'm pretty happy to be able to talk about this stuff. I'll give you a little background about how we're here. There's two parts. One of them is this kind of bigger project that I'm going to talk about right now that Rachel Johnson is really the tip of the spear of at NOAA Fisheries. But the other part, how this really got integrated with this group, is Vicky Fu, who is a 4th grade teacher here in Woodland, asked me to talk to her students just about fish and floodplains and some of the work we're doing. And we quickly realized that there is interest in both what we're doing here, participating with the salmon in the classroom, and then the opportunities that we have to kind of integrate them as not just having growing salmon in the data or salmon in the classroom, but having real useful data come out of it to help the managers of these resources. And so we're working with the UC Davis School of Education and their citizen science program to develop a couple more things that we think might be useful for the teachers and resources for the teachers, but also giving the students something that they can see that how their data is actually being used to make management decisions. So that is how we got here. It's super fun. And I hope that everybody has lots of questions by the time we get through here. And what I'm showing is a pretty high level, the 30,000 foot view of thiamine issues that have recently shown up in the Central Valley. And this is a part of a talk that Rachel had given at the Bay Delta Science Conference that I cut down. It's still probably too long, as I warned Richard that I can talk longer than I should be allowed to. So I'm going to kind of go through this pretty quickly, and then we'll open it up at the end for questions, and I'll probably be going back to some of these figures in here that we can talk about some of the issues with thiamine. So with that, I will start rolling through here. And this is a group that is probably the most impressive group of people I've ever worked with in my life. This is a large group from many different agencies, many different universities, and everybody here is a part of this. And it's one of the most copacetic groups in that everybody works really well together here, and it's a lot of fun. And I think without this group, we certainly wouldn't be where we're at. And if you look at the agencies across the bottom and the organizations, it's pretty impressive. We have retired people. David Honeyfield he is so passionate about thiamine. He shows up to all these meetings just because he can. And so I really appreciate everybody here and what they bring to it. But I just think it's worth recognizing this group as a whole. So thiamine, why is it important? And how does it work with salmon and the ecosystems that are happening? So salmon are one of these really interesting animals that are an interesting link between the marine environment and the freshwater environment. You know, for the most part, we only really see them in the freshwater environment unless they're being caught on a boat in the ocean and ultimately ending up on a dinner table. But most of the times when we see salmon, they're either spawning in the freshwater and ultimately dying and fueling the ecosystem. So this really cool link between the freshwater and the marine environment. And because of that, they are subject to a wide range of issues that affect their entire life history. And recent work has been done looking at thiamine and in ecosystems in general, and it has been described as the #1 emerging stressor for global conservation. And I'll talk a little bit about that. We're going to talk just a little bit about some of the reasons why thiamine is there. But I just wanted to throw that out there at a high level, that this is something that's relatively a big deal that oftentimes many people hadn't heard of. I've never heard of it before we started this project. And the more we look into it, the more we realize there's people doing this in the Great Lakes, in the Baltic, in Alaska. And so it's one of these things that is pretty important. And so why is it important? Thiamine is basically just vitamin B1. It's something that we can't make. Most older animals don't make it, or more advanced animals don't make it. But what happens is that it's synthesized oftentimes at the bacterial level, and it's incorporated up through the food web. And so if you don't have it, though, you have a lot of problems. And so in fish, that's displayed by a loss of equilibrium, swimming in a spiral pattern after they hatch out of their eggs. Oftentimes they're very lethargic. They don't do very well. They oftentimes have internal bleeding. They have lots of neurological symptoms. And that's not good if you're a fish in the world. You can see here on the pictures on the bottom is the picture on the left is a healthy, what we call alevin. You can see its yolk sac is it's one color. If you look on the picture on the right, you can see how there's those like red hemorrhages or like bleeding internally that's happening with these fish, and they don't swim, and they oftentimes lay on their sides in the tanks, which I know from some of the teachers in this program have seen this, I think. We'll get there though. So what I want to do is I want to talk about some of the work we're doing, how we got to where we're at, and I want to kind of finish this by bringing together how I think that the Salmon in the Classroom program and the thiamine program has an opportunity to work together and actually collect really important data. So we'll get moving here, and I will do my best to stay on target. So what we learned was that in 2019, fish came back thiamine deficient. The salmon in the hatchery were having problems. Thankfully, we have people like Kevin and Mark Atkinson, who I saw was on here also, and that's solving these problems and understanding what are the problems with the fish in the hatchery. But one of the things that is unique is that the winter run fish are an endangered population, which means that they're holding on by a thread. And we were worried about when they came back from the ocean that they were going to be thiamine deficient like the fall run before them. And so we devised an experiment to where we would actually treat them with thiamine before they laid their eggs. So basically what we were doing was providing prenatal vitamins for the female salmon. And we wanted to see if we could actually affect how the thiamine concentrations in the eggs, if that was something we could actually manage for these fish that spend some time in fresh water before they spawn, and whether they would be more successful than the fish that were not treated. So here is the injections that were happening. The fish on the left here is being injected with a thiamine solution before she spawns. And then when they were spawned, as we collected eggs, they were sent over to Jacques at SUNY Brockport, who analyzed those eggs for thiamine. And this is what we got. We had data that came back right away, which is, you work with somebody like Jacques, it's pretty impressive. I don't think he actually ever sleeps. But one of the things that we found is that the fish that weren't treated with thiamine had very low thiamine levels. And the fish that were treated with thiamine were very elevated in their thiamine values. But what does that mean from a fish, after, for the eggs of these fish after they spawn, was that we went and we took the eggs, and this is something that many of the teachers here are probably very familiar with, And we hatched them in these individual trays that you can see here. And we wanted to see what was the survival rates of fish with different levels of thiamine. And we found that about 50% of the fish died if they had a thiamine value of 2.7 nanomoles per gram, and about 5% of the fish died if the values were about 5 nanomoles per gram. So there's definitely thresholds there that we saw, and so that we realized that if we can use these numbers to sample the mom's eggs when she lays the eggs, that we can get a good idea of what sort of survival we could expect from the population in general. And so how does this affect the wild fish? And so that is a good question, is that we don't really know, in that all the fish in the wild are almost impossible to sample. But what we can see is that when we took our treatment or our control fish, the ones that were just injected saline, is that a lot of them had values that were relatively low and would have expected to see mortality at the egg level. And so there are several problems of having low thiamine for a fish. There's the ones that we talked about as far as they don't hatch well, they oftentimes lay on their side. But there's also some of these things that are sublethal effects, that they don't die right away, but they oftentimes later in life have the potential to be have poor immune systems, so they don't fight diseases well. They're more susceptible to pathogens. But there's also neurological problems and metabolic problems for these fish, that they just don't really, they don't behave like salmon should. They might not avoid predators as well. They might not be able to get food. They have slower growth. And so because of that, we've realized that this is a real important issue to understand what are the actual causes of these low thiamine numbers. And so we took those eggs that did survive but had low thiamine levels, and we did a bunch of experiments with them at UC Davis. This was Heather Bell as a graduate student at Davis, and this is the work that she did over the last year, which you can imagine with COVID, trying to do these emerging sciences is a very character building experience for anybody, let alone a grad student who's trying to make their way through this process. And so we tested thermal maximum, basically how much temperature can these fish handle before they die? How well do they rest normal? Does their metabolism work right? Are they willingness to be bold and explore new environments, or do they do poorly with predators? And so we are still working on some of that data that Heather is getting back in, but these are the things that we did over the last year. And so one of the things that we realized is that, similar to 2020 for humans, 2020 was kind of a pandemic for salmon also. And understanding how the ocean has changed and what are the causes of these thiamine deficiencies was one of the things that we were really curious about. And so when we looked at the population as a whole, we realized that there are quite a few fish that were coming back that had low thiamine levels that had the potential to have very low survival from those years. And so we wanted to think about what are the reasons that are causing this. And you can see the title of this is, Are Anchovies the Villain? Most people don't think of anchovies as a villain. A fish that lives in the ocean, it's only about 6 to 8 inches long. But what's interesting is that they have something in their stomachs it's an enzyme called thiaminase. And that enzyme breaks down thiamine in the animals that eat it. So when salmon are feeding on a lot of anchovies, they're eating a lot of thiaminase, and so their thiamine levels are getting lower and lower the more anchovies they eat. And you can see what happened in the anchovy distribution on the left-hand side here between 2017 and 2019 is that all of a sudden there was a boom population of anchovies in the ocean. And all of a sudden we started seeing at that same time that the thiamine values in the fish started becoming lower as well. And so if you look at all these causes of the anchovy, what we call the anchovy regime, is that 2019 stood out as a very different year compared to the rest of the gut contents of salmon that were caught in the ocean. And so when you go and look at these gut contents, which is a picture of the upper right-hand corner, that is a salmon's stomach, and it was full of anchovies. So they were just gorging themselves on anchovies in the ocean. And ultimately what happened is when they would come back is they would have these low thiamine levels. And one of the things that we were asking ourselves is, how can we see this ocean food web that is recorded in time? And one of the things that we're doing is with this project is we're reconstructing their food web using the eye lenses of the fish. Here you can see one of our high school interns is pulling fish eyes out of dead fish. And we can look at the lens of those fish and they grow like an onion. And so if we look at those layers of when they're in the ocean, we can see whether they're eating anchovies throughout their whole life or just at part of their life, and how that affects their thiamine values. Because for fish that come back, we can actually sample their eggs, look at their thiamine levels in their eggs, and then look at their food resources from their eye lenses. And so I'm trying to do this as fast as I can. I did that super fast. And I do realize that there was so much information there. And I want to save the Q&A. I want to save that for the Q&A at the end and answer your questions that were coming. I imagine you do, because I really did fly through that. But I wanted to just kind of get a baseline there set and set this up for Kevin, who is now doing the what do we do about part of this, and what are our options, and some of the work that he's been doing, because I think that the opportunities that we see going forward is really in the work that Kevin is doing. And I'm going to let him, oh boy, am I still here? All right. It's different. All right. Am I still sharing?
Yeah, Dr. Jeffries, you are.
Let me see. Oh, there we go. I got it. So with that, I wanted to kind of let Kevin now talk about now what he's doing. And then I also have, as we go through questions, I have a bunch more slides at the end of this talk that we can kind of go into, but I wanted to let Kevin give the science of what he's doing and then move to the discussion afterwards, because I'd rather have that discussion, the back and forth, I think, is where we get to the questions that you guys really have and what we have as well. So with that, I'll hand it over to Kevin.
Yeah, thank you. Real quick, if you have a burning question that you can't write down or you can't remember, stick it in the chat and we'll come back to it after Dr. Kwok's all done. Go ahead, take it away. Oh, I think you're still on mute. Dr. Kwok, I don't know if you're going already, but you're still on mute.
Okay, here I go. So let's see. Can you see my screen by chance?
Yes.
Okay, that was a great overview by Carson, and I just want to expand on it and talk about what we're seeing at the hatcheries. So I'm going to start by giving just a really brief overview of the Fish Health Lab. So the Fish Health Lab provides support to 22 state hatcheries, of which 9 are anadromous. I just want to remind you that anadromous means any fish that is born in fresh water, goes to the ocean, matures, and comes back to spawn in fresh water. So we have 9 of those. We also help any registered aquaculturists. We investigate wild fish die-offs. We also review certifications for the importation of live sport, bait, and food fish. So our hatcheries are spread throughout the states, and they're all the way from Southern California up towards the Oregon border. And the furthest distance between our 2 hatcheries is about 750 miles, so quite a big distance. And so when we get a call that one of the hatcheries is having a die-off or some disease issue, we're basically a traveling laboratory. We pack up our microscopes, plates. We can take samples for virology and other tests if needed. Again, in the state, we have 9 anadromous hatcheries, and I added the symbol for the U.S. Fish and Wildlife right below Redding. And they run 2 hatcheries in that area. One is dedicated to the winter run Chinook salmon. As Carson mentioned, those are endangered species. And they also have another hatchery, the Coleman National Fish Hatchery, that raises fall run Chinook and steelhead. But if you look at the anadromous hatcheries, they're more focused in the Central Valley and north. So we don't have any down south in Southern California. At these hatcheries, the state hatcheries, that is, we can raise spring run Chinook, fall run Chinook, coho, and steelhead, but they don't raise all 4 species. So one of them can raise all 4, but some of them just raise a single species. So it depends on which hatchery that we're talking about. In fall of 2019, September, at Feather River Hatchery, we actually identified a novel fish virus. This virus was first identified in 2015 in the Eel River, and it was the first time seen in California then, but in 2019, it was the first time that we've ever seen it at any of our hatcheries. And through genetic sequencing, we determined that it was a flavivirus. So this virus was identified, cultured, and they did some trials with some juvenile salmonids. And it turned out that it didn't really cause significant disease. But then again, it was still relatively new and new to the state. So fast forward a couple months, December 2019, we began to see early mortality syndromes in Chinook salmon fry. These losses were mainly in our Sacramento and San Joaquin River System hatcheries, but some of the biologists for the state, for our department, were also running in-river traps, and they're collecting fry as well. And they started to see similar behavior and losses that they were seeing in our hatcheries. So again, the focus was just in this area, and you can just see how they have a similar drainage, that their water system empties out into the delta and through the Golden Gate. Our northern hatchery, 1, 2, and 3, they're a different water system. That's the Klamath Trinity River system. And number 5 is our Warm Springs hatchery, and that is the Russian River. So the Central Valley was very separate from the other hatcheries, and we didn't see the similar signs that we were for the Central Valley hatcheries. Some of the disease signs that we were witnessing was inability to remain upright, ataxia, loss of control of body movements, corkscrew swimming patterns, lethargy, excitability, elevated mortality. Just want to show what that looks like. And this was Feather River in December in a raceway. And so whenever you see fish on their side, it's never a good sign. And you can see there's quite a few of them. It's kind of hard to see, but on the bottom, there's another layer of these fish too. So with any die-off, we start an investigation, and that usually starts with the necropsy. A necropsy is basically an autopsy for animals. But after we go through a gross examination, we also include bacteriology, virology, histopathology, and we just look for any signs that could point to what's causing these issues. And I mentioned the flavivirus originally. So the returning salmon, their ovarian samples, the fluid around the eggs, was positive for flavivirus. And then their offsprings a couple months later were showing these disease signs. So at that point, it was suspicious, and we thought it may be related to the flavivirus, but testing for the flavivirus ended up being negative. So at this point, we're kind of at a loss, but we had a colleague, Dr. Scott Foote, who's a leader at the California Nevada Fish Health Center, was saying that he was testing for thiamine deficiency. So there's thought that thiamine may be the cause of what we're experiencing and seeing. And so he performed a bath treatment on sick fish and saw really significant improvements overnight. So that was really encouraging. So at that point, we took this information back to our hatchery, and this time we're at Nimbus Fish Hatchery, which is on the American River in Sacramento, and we collected all these sick fish from their tanks. And at this point, we divided them into 4 groups, and we put some thiamine powder into the water to see how they would also respond. And at this point, we tested for different times. But let me show you the fish. You can just see how most of them are laying on their sides. Some are swimming in a very patternless, aimless way. So again, we divided them into different treatment groups. And overnight, the next day, just like Scott Foote, we saw significant improvement. And so this first group is an untreated group. And again, you just see them laying on their sides, swimming randomly. But the next group is where we treated for 1 hour, and there's just a marked difference. This is 2 hours, and finally the 4-hour group. But you can see that none of the fish are on their sides. They're swimming like they should. They're all in an upright position, and they're kind of schooling and darting around. So they look like they just recovered really well from this treatment. So there are other types of treatments. We just talked about the thiamine bath for fry. And as Carson mentioned, there may be some secondary effects from low thiamine levels. And ideally, you would want to treat these fish, or even better, these eggs, as early as possible. So the thiamine injections of returning adult salmon is a great way to get these eggs thiamine early on. And so we'll just talk about these different treatment methods really briefly. So Carson already kind of touched on the injections and what they did with the winter run Chinook, which is an endangered species. We like to do something similar with our spring run Chinook at Feather River, actually starting this Friday. And I just want to point out, like, the winter run Chinook salmon was a great candidate for this study because they actually collect some of these fish and bring them to the hatchery where they can hold them. And so they injected them and held them for about a month before they spawned them, and they were able to measure those thiamine levels. Our spring run have a different life history. They come in relatively early to the river system. They come in late May through June, and our department tags these fish. So we have a great opportunity to inject these fish with thiamine, and we're going to release them, and they hold in the river for a couple more months, and then they spawn in the fall. So when they come back to spawn, they'll be marked, and we'll take some eggs to measure the thiamine levels. And just like the winter run Chinook, we'll also have a control group that gets injected with saline, and we're going to do a comparison between the thiamine injected and the saline injected for the spring run. However, most of our anadromous hatcheries, they raise fall run Chinook, which has a different life history. Once they mature in the ocean, they kind of shoot into the river ready to spawn. So they don't spend any time holding, waiting to spawn. So as they enter the system, they're ready to spawn, and there's not a great opportunity for us to inject them. So we have to find different ways to treat these group fish rather than trying to inject them. One way is a thiamine bath of eggs after they're fertilized. So it's this period called water hardening, and the eggs finish up hardening their egg shell. And so at this period, we can incubate them in a thiamine bath. And this is a method that's used in other hatcheries, like in the Great Lakes and even on the East Coast. But for us, it may not be an ideal method. And one reason is, once these fish eggs are fertilized, we typically disinfect them. So we put them in an iodophore solution, and that removes any pathogens on the outer surface and prevents them from trying to get into the egg. So if we do this thiamine water hardening period, we're going to skip over this disinfectant period. And that comes to the fertilization with thiamine water. So this is adding thiamine right at the fertilization stage. Some of the work has shown that it will definitely bring these fish out of a critical area. As Carson mentioned, that zone is 8 nanomoles per gram. And we were able to show that a high dose of thiamine in the water will pull these eggs out of any deficient critical levels. So this is really promising. It's a very fast and easy way and a cheap way to treat these eggs. So at this point, I just want to show you how fast it is. That water is what they usually use, but it has thiamine this time. They add melt and they just swirl it, and that's it for the treatment for thiamine. So now they just move on to their regular protocol. They rinse the eggs, and then those eggs go into a disinfectant. And so that takes about 20, 30 seconds. So it's a great way to add thiamine without having to use more resources or cost for this process of treatment. So with that, I just want to say a salmon, it's a hard life. I mean, there's so many different ways that a smolt can be affected, or a salmon, I should say. And we're talking about such an early stage in life, at the egg stage, where they can become deficient, and that affects them throughout their whole life if they even make it past the fry stage. And once they get in seawater, there's all kinds of other things they have to face and deal with. And the picture in the right-hand side, bottom right, it just shows all the predators that like to eat salmon. And I'm one of them. I love eating salmon. And I just hope when I come back in my next life, I am not a salmon. But the lower left also shows the natural life history of a hatchery salmon. And unfortunately, we have built dams for various reasons, but it has blocked salmon from their natural spawning grounds. And so these hatcheries are here to help mitigate the loss of those spawning grounds. And so when they're taken into the hatchery, we're looking after them. And we also help with that early rearing stage of their life. And so it's my job to give them the best chance of survival once we release them. And so I just want to thank all the people that helped get us to this point. It's the same group that Carson mentioned earlier. It's been an incredible group to be a part of. Any questions?
Dr. Kwok, Dr. Jeffries, thank you so much. And yeah, I know there's some burning questions out there. Please feel free to get on the mic, put it in the chat if it's more comfortable for you. And yeah, let's get in it.
This is Ethan. I'm in the Bay Delta region, and we do not deal with salmon, but we have had issues with rainbow trout swimming in circles. And I'm wondering if you feel that thiamine deficiency could be the issue that we're looking at.
I think it could be a potential issue. We do have some hatcheries that have shown their steelhead have had low thiamine issues, but the trout you're talking about, are they being caught, or where are they from?
I'm sorry, this is with the Classroom Aquarium Education Program. So mostly we deal with rainbow trout that are sourced out of Mount Shasta Hatchery and then transferred to Silverado Base. Okay. We do deal with some steelhead, about 120 classrooms, a very small percentage of our overall from the Warm Springs Hatchery, but I think our swimming in circles has been primarily rainbow trout.
I think it's less likely to be a thiamine issue. The broodstock have a balanced diet. They're being fed a commercial diet, and I know they fortify with thiamine. And so I believe it's very unlikely to be a thiamine issue.
Do you want to take a stab at what you think it might be?
There's a couple things that I've seen. I mean, there could be a water quality issue. There could be something called coagulated yolk, where we've seen their development is not normal and they can curl up and swim in circles. It could be a pathogen issue. So there are a couple different things that it could be.
All right.
But my first guess would be a water quality issue.
All right, thank you. It's helpful.
I I also see the question that John Duran just asked about what is the outlook for the wild salmon? And I think that that is a great question. And I also think it's the reason why I ultimately got interested in working with the Salmon in the Classroom, or the Fish in the Classroom project, is that this is one of the few groups, like Kevin was talking about, that there's the opportunity to treat at fertilization. But the other group of fish that never got treated prior to Kevin, if Kevin wasn't so fast with solving problems, then this would be a great opportunity. But the fish that go to or have gone to the Salmon in the Classroom program weren't treated. And so even if they got sick and they were displaying some of the symptoms that Kevin had shown, was that might be one of the control groups for the wild population, because there were in 2019 fish that were being collected in the traps, the fry traps, the out-migration traps, that showed some of the wild populations, but there weren't any real numbers associated with it. And when Vicky, who I think is on the call, sent me, when I was talking to her classroom, we had this discussion about it. She's like, "Oh, can I show you a video of my fish?" And she showed me the video of the fish, and it looked exactly like the video of our thiamine deficient fish that we had in the lab. And that was when the light bulb went off of thinking, wow, if we have this group of fish that never gets treated, and we're able to collect the data for them that go out and who survives, how many survive. Obviously, there's lots of things that can, lots of reasons fish can die, as Kevin was just talking about. But if we were able to have a pretty simple questionnaire, if there's even an app that students and teachers just filled out for fish that exhibited a couple of these pre-distinct signs and symptoms, then it's one of the things that we can get an idea of what's happening. It's not exactly a wild population proxy, but it's pretty close, and it's something that we can think about using. And we just put together a proposal that incorporates development of something like this with the citizen science program at UC Davis, and trying to think of an easy way to do it and still provide data and also some explanations. I mean, one of the things that, as I've done this a couple of times, is a couple of teachers have been like, boy, my kids were so disappointed when the fish hatched. They laid on the side of the tank and kind of swam around in circles like Kevin was showing, and they thought they had done something wrong. Mm-hmm. And the reality of it is they didn't necessarily do anything wrong. It might have just been that there was a problem with the fish, and that data can be really useful as it kind of goes up into the management area. And I think it'd be really fun to try and coordinate with some of these to kind of get an idea of what's happening with these types of situations.
And it was also really interesting because for our hatcheries, not all of the fish developed thiamine deficiency. So we had lots that were specific to thiamine deficiency, and there are other lots on either side that were not. So I was really curious who was showing up as thiamine deficient. There was no real pattern that we could find.
We got our eggs from the Orville hatchery, and the batch that we got, the lot number, I think it might have been 3 or 4, definitely had the thiamine problem. I only had 4 classrooms of teachers. Every single one of them had problems, and a higher number than we've ever had. Occasionally we get one spinner here and maybe one there. Every single teacher was contacting me saying, my fish are doing this, my fish are doing that. What am I doing wrong? And so I started contacting the hatchery to find out what's going on. And I started hearing about this thiamine deficiency in the anchovies. And that's all I knew. Now I have a much better idea. And I'm one of those data collectors. Besides the teacher sending in the completed permits, they send them to me. I take all the information out and I do a data chart. So we see what's our success level, how many fish survived, and what are some of the issues that we had with them that didn't make it. So I can expand on that data, and I send it into Richard every single cycle.
Awesome. That's wonderful. And that is exactly, I think, what how we'd want to do that, is that if we could formalize a way that has some of these descriptors of symptoms, it's an easy way to upload, and it provides some sort of context to the students. I think that's one of the things that it hurt me when teachers are like, oh, all my kids feel like they killed their fish somehow. Mm-hmm. And, I think that being able to, provide students with the opportunity to be like, hey, these are some possible reasons why it could have happened. and developing something that is simple enough that the folks that we're working with at the School of Education, this is what they do, is making things that are easy, like walk through steps to understand. That could be a couple things, but also provide that data back up in a way that's useful as well. And so kind of having a two-way flow of information, that it comes with like maybe the idea is to develop like a one-page thing that go to the teachers that might explain some of these things, but also the way to send that data up quickly and see it as well.
Yeah, thank you. And I think that's what we got to do. We got to figure out those channels. And so Gail, I know you had a comment, and then Sandy had her hand up, and then Vicky has her hand up. So we'll go in that order.
Well, this is Gail Davis, and I work with the classroom program throughout Central California. So we got our eggs from the Merced hatchery. So Carson, the way you were talking, it sounded like you were more interested in the eggs that would come from hatcheries that don't treat. I'm assuming if one treats, they all treat, or how is this going to be implemented? So
That's a great question. I would almost leave that to Kevin at this point, because I don't think that, and correct me if I'm wrong, Kevin, but there isn't a full treatment plan right now, is there? Or is that's kind of what's happening, is we're figuring out what the scale of the problem is and what are the solutions to go forward. So I will let Kevin answer that one.
Right. When this first occurred, we had hatcheries treat most of their groups. Now that we have a better understanding what's going on, some hatcheries want to wait to see if they get any data before doing some of this treatment. Because right now our standard of treatment is to expose these fry. And now we're looking at fertilization treating at fertilization. So we haven't worked out all the details, what we're going to do with hatcheries this year. That's something coming up really quick. But there would probably be some type of standard, or some hatcheries not treating some groups to see if there's any difference too. So you could get probably an untreated group if you just work with a hatchery.
I would say the other thing that we're trying to do, Gail, is we're trying to develop a surveillance program with the commercial fishery, is that being able to sample females prior to coming into the ocean for spawning, is that understanding, if you go out and you sample 1,000 fish, looking at what their thiamine levels are prior to return, we might get an idea of what sort of magnitude of thiamine deficiency at a population level we'd be coming back with. And so the thing that's kind of cool about this is we are a year into this problem, like this program. And honestly, I have never seen a program move this fast. And part of it is the amount of people who are on, is that there's all the agencies, there's a serious brain trust. I feel so undergunned when I show up to these meetings from an intellectual standpoint, and that there's a lot of brain power that's going to this right now. And I think that it is evolving quickly, and I think that it will continue to change over the next year, because we're a year in to see kind of this major shift in management.
well, I'm just curious. We did have a thiamine deficiency, seemingly so, last year. I mean, not this previous year, because not enough salmon came home that we even got eggs. But the year prior, we did have eggs, and we did have problems. And same thing, teachers are asking me about this. So, when it came out that it might be thiamine, then it was, springboarded into a whole nutrition lesson. But also, if the conservation of the salmon isn't enough for you when you're doing your work, you can always consider the thousands of children out there with puppy dog sad eyes that their fish are dying.
That is what got me.
There you go. There you go. So, and before I leave, just wondering, would either of you be willing to do like a Zoom talk or something with a class at some point, or with teachers in the Central Valley?
Yeah, I mean, for me, I obviously love talking with kids. I think it's the most fun thing probably in my job. I'm just really a big kid anyway. And I think that figure out a way to to do it for 100 classes is starting to push it.
Yeah, no, I don't
but
think we'd have that many.
And so
but-
figuring out how to make time work. I'm always into trying to make it work. If there's groups of classes, that always makes it easier to kind of combine. But yeah, in general, I'm pretty amenable if I can find the time to make it work.
Okay, thank you.
I'll let Kevin speak for himself.
I'm in the same boat. I would love to do this, and if it works, I would be happy to be a part of it. I've been to Merced. I've seen the kids go through there too.
Abby?
2 years ago when they were having thiamine deficient issues. And yeah, I would love to support that area.
Well, that'd be great if you come down to Merced, because it's such a small, intimate kind of hatchery where it's really you're there. I mean, I have to warn the kids that you might get splashed with blood and guts, and that just makes a rush into the building because they want blood. So, but that would be perfect if you guys could come out, and maybe even if it's not, we could do interview you and just let teachers post or listen to that video. It wouldn't have to be 100 interviews, which would probably be helpful. So thank you for everything. Thank you for being here and helping us understand what's going on. So.
Yeah, thank you, Gail. Sandy, you're next up, and then Vicki, you're after.
Yeah, hello everyone. I really love the idea of a common data set, a data form, data collection, especially coming from the high school environment. I'm a science coach in Lodi Unified and had used the program for a long time with my biology and AP environmental science students. And Catherine, who's teaching the same courses now, is here. And I think having a really formal, nice data sheet that maybe even has like a UC Davis stamp or a CW FW or CDFW tag on it, something that's very official looking, kind of takes it to that next level for students that they are collecting data for a higher level, and having that common thing that then all of our schools could have across, and we could really have that shared data point. So I want to champion that idea for sure. And that was pretty much what I wanted to say, because I'm kind of the point of contact. So me developing something wouldn't help across the valley. So just throwing that out there. And then Catherine had a really good note in the chat about tagging the fish. So I don't know if she wants to expand on that or just take a look at her note there.
I could probably go for both of those real fast. Exactly what you said about the data sheet is what we're talking about. And also having the citizen science group they've done this for lots of different things. And so they had an idea of how binning it by different age groups. There'd be like a middle school, high school bin, a 3rd through 6th bin and a 1 through 3, whatever. They had those bins of that. They would be easy to fill in for each of those things that they've done in the past. And so, yeah, I think that would be the goal. And I think it'd be coordinating with CDFW. I think NOAA would also be involved. NOAA's, as Rachel Johnson, who is really the tip of this spear. She's a NOAA employee and a UC Davis employee. And so that is the thing that we want to do, is make it so where it would be simple, it would be effective, and it would be useful. And I think that's one of our goals. And about the tagging, it is really hard to tag small fish. I will, as somebody who spends a lot of time tagging small fish, it is not an insignificant amount of effort. And the hard thing is the recovery when they come back. If a school has, let's just say 35 fish, the odds of finding one of those are so small because you have to look for them as adults. That's the challenge. It's one thing to let them go. It's the next thing to look at every adult salmon in the commercial fishery, in the recreational fishery, in the spawning ground surveys, in the hatchery. It ends up becoming really difficult. But I agree that having ways to tag fish is really interesting, and we're working on some ways, whether it be genetic is one way that we might be able to get at some of those, is doing some of the parental-based tagging where we could basically do ancestry for fish. And we have that capability of doing it pretty cheaply. And I think that's something in the future. It's not something we have right now, but it might be something that we can potentially do in the future. So
that is so cool. Thank you, Sandy. And Vicki, I know you've been patient. Did you want to jump in?
Oh, yeah. Hi, everyone. So I teach fourth grade, so I'm kind of looking at this from like the littles perspective, and I think that just hearing all of you guys who are interested and have kind of their hands in the pot, I think it'd be awesome to have some like, I don't know how to get this going, but like some kind of network between other, school sites that might have salmon in the classroom, the same run, and kind of monitor that as well. I think that would be really awesome. Another lesson that I'm thinking about from this conversation is, this is science in real life as it's happening, right? We have an issue, and here are real scientists reaching out to different agencies, government agencies, and all of that. And so, kind of linking it back to, how do we as a society kind of look at issues and solve them together? So I'm super excited. I'm so glad that our conversation led to this, Carson.
I am too, Vicky.
Me too. Me too. And I feel like this conversation is just going to keep going and keep evolving and growing. And so we only got about 4 more minutes. I want to be respectful of your time. I think there's one other person with the hand up. Who is that? Is it Brian?
That's right. So I'm Catherine. I'm just I got home and I logged in on my husband's computer. So I'm showing up as Brian Matthias, but I'm Catherine at Tokay High School, and thank you so much for doing this. I just wanted to say that before we log off. We're new to the area. We're new to California, not necessarily new to fish, but new to salmon in the classroom. And I did it this year, and kids had a lot of fun. And I'm always looking for more resources to adapt to high school. So I'm at the high school, and I found a lot of resources for elementary or middle school, and to kind of scale that up. It just takes time. So if there's anyone out there that, I guess if you think of something that you would really like high schoolers to know about when they graduate high school about salmon in the Central Valley, or part of that story that you'd want to tell, you have a platform. And if there's something you'd like to share, that'd be really great. And it might take some time to think about that. So if you think about that later, maybe we can set it out later.
No, and I will jump in as a shameless plug, is that and for those of you teachers, is for the Common Core standards for the phenomena, which I imagine is a term that you all recognize, is we have developed some of the phenomena, what are they called, modules, I think, for a variety of things, and we did one for floodplains and salmon. We developed one for, elementary, middle, and high school units that I got to be part of as part of the Creek project, or the Creek network, which I think some of you guys probably know these terms better than I do, even though I am married to a teacher, so I should know this. But I think that there are some of the resources that I know Crystal Howe at the San Diego County of Education she's been kind of the hub of those phenomena that we've put together. So I would reach out to her as an opportunity and as a resource for some of these salmon-based things that we've put together in the past.
Could you say that name one more time?
Oh, Crystal, Crystal, Howe, HOWE, and I think if you go to Creek, CREEC.org, it might be there. My wife is probably hearing this and just like shaking her head at how I'm giving bad information. But there were a series of phenomena summits that were held throughout the state, where they brought in folks and developed different phenomena ideas and modules to fit in within the NGSS standards. So I know that there are some of those resources out there, and I would point towards that first.
Thank you so much.
So
thank you.
Kathy in Lodi was asking a couple questions. One of them, the first one is Mokelumne Hatchery. Do they have any issues? 2 years ago, they did have issues and they treated their fish. And then the other question was asking about treating their fish in tanks, the fish in the classroom. So this is opening another can of worms, a very different can of worms, just because thiamine truly isn't approved use in animals. So when we're treating our fish, it's under an off-label prescription. And so it's done with veterinary oversight. So treating in the classroom, I would say, would be a little more tricky and difficult to do just because of the logistics and the paperwork and liability involved.
Thank you, Kevin. Thank you for. I wanted to get to those chat questions, so thank you for being on ahead of me on that. There is what we have Gail with one more question. Do we have time for one more question, fellas?
Yeah.
Yep.
Okay.
So my question is to you guys and you, Richard. So this is all wonderful stuff, but it's an ongoing saga. So how do we keep in touch? How do we know the latest and greatest? How can we keep this going?
That's a great question. I hope we get our proposal funded. Would be the first thing, because we do have this explicitly as part of it, is the outreach and coordination with this program. And so that's hopeful.
When will you know?
Hope is not, I was about to say, hope is not the best strategy for making that happen.
Yeah.
It's a strategy, but it's not the best strategy. So I don't really have a plan. I think that I do agree that keeping this communication open is very valuable. And I'm not sure about the best way to do that is part of the program. But I think that, that we're at the, Richard and Kevin and I have had one date at this point. So this is our second date. And then this is our first date with everybody else. And that, we're just trying to figure out how this relationship is going to work. And I think that, as this has been really informative for me. And I think that figuring out that there is a need for this communication strategy. We spend so much time communicating to other scientists and management agencies, but we need to figure out how we do this well. And I hope that this is a start of that. I don't have an answer for how to do it. Richard, I don't know if you have any ideas.
Yeah. So what I'm going to do, I know for sure, is I'm going to be working with other coordinators in the state, with all the sponsors and the coaches and leaders that deliver the fish to the teachers, to develop that data sheet. It'll probably go through a number of iterations. We'll probably run it by you, Dr. Jeffries, by you, Dr. Kwok, try to reach out to NOAA, really kind of vet that data sheet so that it's useful and provides that meaning if there is a tank failure for those students, and just really just kind of boost the program in that way. That's one way I know that some steps that tangible steps I'll be taking to work with, so that hopefully by next Chinook salmon season in the fall, we have this thing ready and we could use it to at least start collecting data and then beginning to figure out what to do with it. So that's one way we keep the conversation going.
So is there a way that we can, maybe we can, I just want to, it'd be nice to have like a group of communication somehow, so that either it'd be like an email list or something where we're, hearing this, or if we could, hear updates whenever we need them, or every 3 months, or I don't know. But this is like, I have worked for the department now for, I think I might be up to my almost 30 years, 29 years. And it's been, to have biologists talk to interpreters is huge. It's a big deal. And if the wardens would talk to us too, that'd be great. But I work at the LaGrange office, and so I could just, stand at their cubicle. Before COVID, I could stand at the biologist cubicle and give them the evil eye. And then they'd talk to me and get an answer. But this is so fantastic. I'm so excited. And I don't want it to end. I don't want it to just, so I don't want to drop the ball on this, because we've got you now and I don't want to let you go. So,
I think
I will tell you, I was going to say, my goal is to institutionalize this through the citizen science program on campus. That's if we're supposed to find out in the next month whether we get funding or not.
I'm in Merced. So I'm out of the LaGrange. How do I keep it going
Well,
out
yeah.
of the Bay Area,
No, we're in the Central Valley. We're at Davis. We're not the Bay Area.
Well, Sacramento, I'm the poor orphan San Joaquin system,
No,
you know?
Yeah, no, we want to make this a programmatic thing. And I think that's why working with the citizen science group and what they've done is they've developed programs throughout the state that are able to have a communication that goes back and forth so that teachers and educators are getting the information to get the data out of the students. And so that there is a back and forth and that it's done in a way that has been shown to be effective and there's literally an entire institute on campus that is about this. And so they do it really well. And that's who we've partnered with in this. And if this works, if it happens, then I think that's going to be the mechanism. If we don't get it, we're going to have to think of something else. But I think that, that we're interested in how to make it happen. It makes it a lot easier if there's something that's institutionalized and it's actually part of a program. And I think that would be fun. I think it's something that Richard and Kevin and I want to bring Rachel in as well as our education team on campus. And I think that will be where we really bring it together and make it into something that has a more regular interaction. And it's not just the local schools here, but it's something throughout the program, I think is what we want to do.
Yeah, I just want to second that and echo that. I think that is the means by which this conversation, this momentum continues and even picks up pace. So that's my goal along there with you, Dr. Jeffries, is to how do we institutionalize and integrate this into not only the Classroom Aquarium program across the state, but even wider. How wide can we get? And I did want to say, it sounds like Gail wants to go steady. She's looking for an engagement. All right, she's looking for some serious commitment, which I love. And but with that, I appreciate everyone's time. We're a little bit over. Hopefully, this was edifying for each of you. I want to thank Dr. Jeffries, Dr. Kwok. You guys are experts in your field and amazing. And I appreciate your time and your expertise to share with us and all the teachers and everyone that's participating. Thank you very much.
Yeah, and thanks you guys for having this program available. As a kid, it's a pretty cool thing to have fish in your classroom. I do appreciate all the work that goes into making that happen, because it's not just something you do real fast and it's over. It's actually maintaining that for a long time, and I do appreciate that. So thank you guys.
Yeah, it was definitely great reaching out and getting this information. And as Gail said, hopefully this is ongoing and we can spread the word even further, because it's not just local fish issues. I mean, we are hearing from Utah and their fish may have a similar issue. And there's even talk about like alligators, or are we seeing this in whales? So it's such a big issue that involves so many different parts in this world and not just our local fish population or community. So it'd be great if we could keep it going and expand it.
And the other thing too, I appreciate is because in turn, we'll be sharing this information with students who are ones coming up, our stewards of our beautiful planet. They need to know. And the more science we get into them, the more excited they get about science. And that's what's wonderful about this fish program, is there's so much that can go into it and just make them more knowledgeable about what's going on in the world.
Yeah, well said. I think we'll end it with that. Thank you all. And yeah, thank you. Thank you, Dr. Kwok.
Thank you.
Dr. Jeffries.
Thanks, everyone.
Thank you.
All right, y'all have a good night.
All right, bye.
Bye-bye.
Thank you. All right, Rich. Thank you, sir. Nice meeting you.
Yeah, likewise, Rick.
Hey, you give me another email on the next one, okay?
Hey, okay, yeah, we'll do.
Okay, appreciate it. Bye-bye.
All right, take care.